Good afternoon. My name is Olivia. Today we are going over DNA replication, how it works, and why it is important. So what exactly is DNA replication? Well, DNA replication is the process by which DNA makes a copy of itself during cell division. Essentially, it is the process by which a piece of DNA splits and eventually duplicates itself. Before we start, let's examine a piece of DNA so that we can understand all the different parts that make it up.
So at first sight, DNA looks like a double helix with a bunch of bars in between it, kind of like a twisted ladder. While this is a good visual description, what's exactly going on here? Well, the helix strands that you see are made of an alternating pattern of sugar and phosphate groups. So this would be the phosphate and this would be the sugar. These little bars are made of two nitrogenous bases that are connected with the hydrogen bonds, which you can see here, with adenine and thymine and the hydrogen bonds connecting them. Only certain types of these nitrogenous bases can actually connect with each other. Here's a good way to remember it. Adenine, like apples, goes on trees like thymine, while cars, like cytosine, is paired with guanine, like garage.
Firstly, the DNA is unzipped by an enzyme called helicase. Unzipped refers to when the helicase breaks down all the hydrogen bonds that connect the nitrogen bases. Gyrase, another enzyme, prevents the DNA strand from tangling. This whole process is stabilized by single-stranded binding proteins, which help keep the single strands of DNA intact. Next, an enzyme known as RNA primase adds RNA primer to the 3' end of template DNA. This allows one of the original strands to attach to a new strand rather than only being able to attach to the other original strand. For context, DNA is something called a semi-conservative molecule. This means that when it copies itself, one strand will be new and the other will be old. After that, the daughter strands are built. DNA polymerase III adds new complementary bases to the new DNA strand. DNA has two strands called the leading and lagging strands. The leading strand is 3' to 5', which means that all of the nucleotides can be added at once in a continuous strand. However, the lagging strand is 5' to 3', so the nucleotides have to be added in smaller pieces, which creates a discontinuous strand. These pieces are called Okazaki fragments, named after the scientists who discovered them. They are then connected by an enzyme called ligase. Finally, the RNA primer is replaced with DNA with the help of DNA polymerase I.
Sometimes when DNA is being replicated, it can create a replication bubble, which is this. A replication bubble is a region where two replication forks are in close proximity to each other, and it can cause bubbles in the replication DNA. And that's it. To recap, the DNA is unzipped by an enzyme called helicase before another enzyme called RNA primase adds RNA primer, which allows the original DNA strand to attach to a new one. Our DNA polymerase III then forms the daughter strand, and DNA polymerase I replaces the placeholder RNA daughter strand with an actual DNA strand. As I'm sure you can tell, DNA replication is a pretty complicated process. Thank you for watching!